more. Okay, so yeah, so that's where we're going to pick up from, is where we sort of got to this time just before the summer. So the last little bit for year 12 is this sort of evolution, natural selection sort of bit of a topic. So it's quite a short little topic, but basically what we're going to look at is the concept of Darwin's evolution by natural selection, and look at molecular evidence for evolution. So looking at genetics and genomics and seeing how they sort of work together to sort of prove and give some evidence for evolution. So basically, Charles Darwin, you've done about him at GCSE and before, he was basically a naturalist, and he spent a lot of time out in the Galapagos Islands, and he would observe animals. And what he basically noticed, animals and plants, is that obviously we have lots of similarities between different species. We can do that quite naturally ourselves, like we naturally categorize things, because we talked about taxonomy before, like how we group things, and we go, oh well, we can start to group things quite clearly because they have wings. So we say, oh, birds look really similar. Now we know a parrot and a pigeon are different, but actually they still have those similarities. They still have wings, they still have beaks. So Darwin used this sort of information, and he believed that these similarities pointed to a common ancestry, and that the new species formed by gradual changes over the generations. So his thought would be, well, birds all have wings. Somewhere in the past they will have had a common ancestor, and obviously you'll have other birds that are a lot more similar than obviously the huge difference between a parrot and a pigeon. So for example, Darwin looked at finches, and these finches all were very similar, but they had very big differences. Lots of it was to do with the shapes of their beaks, and that was the idea that these new species have formed by gradual changes over generations. So Charles Darwin and someone called Alfred Wallasey proposed natural selection. And this is the mechanism by which the most successful changes are passed on, and that allows organisms to evolve to suit their environment. And again, it's really important to point out here that evolution isn't a active process, it's sort of passive. So whilst natural selection is choosing these ones, the animals themselves aren't necessarily sort of controlling it. It's happening by random mutations, random events. There's a random mutation that arises in the animals that allows them to actually be a bit more successful than another individual. There'll also be a lot of individuals in that population who had random mutations that aren't beneficial, so those ones aren't passed on. That's sort of what we'll look at. So some of the evidence that Darwin had for natural selection and evolution. So Darwin was around in about 1858, and so Darwin was able to formulate his theory without any of the molecular evidence that we have today. So we're going to look at some of them. So he looked at living species. So one of those examples I spoke about was the finches. So whilst Darwin was in the Galapagos Islands, he noticed that finches had different specialised beaks. They all had very different shaped beaks. Some of the beaks were really good and strong for crushing seeds, while others had more delicate beaks that were able to pick insects from holes. But they were all finches. So this is the idea that Darwin then thought, well, all finches could have evolved from one species with a generalized beak shape, and then by that random mutation, one of them's got a slightly different beak. Actually, a slightly different beak, it realizes it can eat this different food. If this one individual can eat a different food that the others can't, it'll be more advantageous because it's got a larger food source and there's less competition. So it's probably going to get mated with, and then that will get passed on along the generations. So that's a general idea where Darwin sort of, what from a living species, one of them where he came from, and it's a whole, rather than just two on this picture, you can get images where there's a whole sort of wheel of the finches, and they've all got these very different intricate beak shapes, and they all basically eat very different types of food. So it goes into that idea of a niche again. So they're fulfilling a niche. They're all specialised, and actually by being specialised, it's reduced for competition. Darwin also got some evidence for evolution from fossils. So the fossil record has revealed many ancient organisms that appear to be transitions between different animal groups. So for example, if we think back, like obviously we have this idea that all life sort of began in water, and then we had animals on land. Well, how did animals go from water to land and things like that? So we have the Ankaphostega gunnari, and we look at this as a missing link. And it has a mixture of fish and tetrapod traits. So if we have a look at it, it's got gills, which obviously is a really distinguishing trait of fish. It's got tail fin from a fish, but it's actually got 4 limbs, which have 8 digits on them and a weak body. It's actually starting to form body with spines. So it's sort of this sort of half fish, half tetrapod. So things like those fossils would start to indicate that maybe there was a missing link. So as animals have transitioned from, organisms transitioned from one sort of environment to another. Darwin also used comparative anatomy. So, and that's actually when you look at the limbs, the skeletons really of living and extinct vertebrates. And you look for the similarities in the bones. One that we've actually really documented really well is horses. But if we look here, basically what's happened is we've got highlighted the humerus, the ulna, the radius, and the carpal. When we look at them, you can sort of see where they sort of started and how they've elongated over time. So this comparative anatomy suggests the organisms are like to evolve from a common ancestor. So if we look at that Ankaphostega, which we had from before with the fossil, it doesn't have a carpal, but it has its humerus, ulna and radius, and the toes. And we look at a lizard, we've got that humerus extending, the ulna and the radius still there, but they've extended. We've got that gap between them, which is very similar to what we would see in a human today. And we've got the carpals. Bird, again, actually quite similar to the lizard, but the ulna and radius is more extended. And then in the human, we've obviously got the extended humerus, ulna and radius, and then those the carpal there. And finally, artificial selection. So domesticization has actually been a really strong sort of indicator for evolution. So domestication of animals shows that animals can undergo dramatic changes over a relatively short number of generations. A big example of this is dogs. Different dog breeds all look hugely different to the point that you wouldn't believe they're the same species, but they are all the same species. So a dachshund, a little chihuahua, they are the same species as, for example, a Great Dane, a St. Bernard, a husky. They're all hugely different, but they're still able to breed with each other. So animals have been selectively bred for centuries, and so this leads to a wide range of dog breeds, for example, or we use this a lot in agriculture, so cows that produce excess milk. So the cows that produce the most milk were the ones that were always bred from. The cows that produce the most meat were the ones that were chosen to breed from, so to increase those favourable traits. So then we've got molecular evidence for evolution. So all organisms share a number of molecular characteristics, which suggest a shared origin of all life on Earth somewhere. So all organisms have DNA or RNA as their genetic material. So we know that there must be something linking them. We know the genetic code is universal, so all amino acids that are made with certain codes in a human will be the same amino acids that are made in a cow. ATP is the universal molecule for energy storage. Any organism, their energy storage is going to be using adenosine triphosphate. Proteins in all organisms are formed from the same 20 amino acids. So not only is that genetic code to make those amino acids universal, the proteins that we make in all organisms are formed from those same 20 amino acids, whether it's in humans, other animals or plants. All organisms have phospholipid membranes, and vital physiological processes follow very similar metabolic pathways in all organisms. And that's really quite important because we're not looking at like those really small physiological processes. We're looking at those vital ones, those major ones, those sort of key to life ones. Obviously, all processes are important, but when we're looking at those like sort of key characteristics, they follow very similar metabolic pathways. So, for example, even just looking at something like mitosis, meiosis, cell death, psychokinesis, anything that's sort of universal. They have very similar metabolic pathways in all organisms. So genomics. Genomics is actually the study of all the genes in one organism. So basically what we do with genomics is we get a genome of an individual, of an animal, or of different animals, and we can actually look at one genome of one animal and compare it to another. And through this, we can establish a relationship. We can look at what do they have similar and what do they have different. Differences between proteins can also be used to establish evolutionary relationships. So there's likely to be huge differences between the genomes of different species. And what can happen is scientists can then compare the genes that encode proteins used in important biochemical processes. So it's like what I was saying before. It's those really important ones. Obviously, there's going to be lots of different variations, but it's these important ones. So these proteins, such as things like hemoglobin, they rarely change. So no matter what animal you're looking at, the proteins that make hemoglobin, they rarely change. And that's because most modifications would affect a key biological pathway. So blood in all animals works in the same way. The hemoglobin in all animals works the same way. So the evolution of hemoglobin in the primates, just going to have a quick look at this. So the table shows a number of differences in the amino acids. In the amino acid structure of the hemoglobin between humans and some other primates, and HB indicates high relatedness. So let's have a look at this. So basically, actually, no, I want to go back to that. No, reset. You're not going to reset now, are you? No, of course not. So basically, this is a number of amino acid differences from humans. So basically what we have here is we've got chimpanzees and humans have 0 major differences, amino acid differences. So what we're trying to basically do here is look at which ones are most closely related. So we've got chimpanzee and humans being 0 with each other. So they would be basically coming off the same branch with each other. So they're going to have a direct common ancestor with each other. So the logical place, to be honest, chimpanzee or human can go either place. I'm going to put them here. They both have a direct common ancestor of each other. So then the next one, with anything slightly different, is a gorilla, and they have one difference. Okay. So here we've got a common ancestor. We don't know what the common ancestor is. We also have this link here. So we would go with gorilla over here. Our next difference is going to be gibbon with 3. So if we look on this phylogenetic tree, chimpanzee, human coming from the same evolutionary ancestor. We've got gorilla coming off that. The next one, well, actually, if we go across, we've got this branching out down to here. So this is where our gibbon would go. After gibbon, we've got a rhesus monkey. So if we follow this tree now, okay, so this is where our next common ancestor is. Okay, well, it branches down there. This is where our rhesus monkey would go, followed by squirrel monkey, which would go at the top, and then lemur at the bottom. And so that's how you can make this primate evolutionary tree. And then we would say, okay, so the lemur is the most different from the chimpanzees and the humans. Then the squirrel monkey, the rhesus monkey, the gibbon, the gorilla, chimps and humans have got the same amounts. So let's just minimize that a minute. Let's open that. So let's carry on and look at natural selection in a bit more detail. So we're going to look at Charles Darwin in a bit more detail and his theory of natural selection. I'm going to look at the conditions needed for evolution. I'm going to really talk through this for exam questions as well, because there is really key points when answering exam questions on natural selection. Basically, it has to be logical. So it's like at GCSE when you have like those 6-mark questions, and they'd look for that logical coherence in your question, in your answers even. But there also needs to be the timeline in it. So it's almost like you're writing a story, and it's almost best just to learn sort of the process of, well, I always say this first, followed by this, 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 and this. And I'll go through it with you in a moment. But that's one that's really key. There's basically a set way that they want you to answer these questions when it comes to natural selection, so that's always good to know. So yeah, the conditions needed for evolution, variation, and mutagenic agents. So again, Charles Darwin was born in 1809 and was alive till 1882. We know he's best known for his theory of evolution, and that was a theory to explain how species change over time. As we said before, he noticed that some species or individuals of a species are more successful than others. He proposed that evolution took place through natural selection and sexual selection. Now, that's really important that we actually have both included there. Now, we basically categorize them into one and call it natural selection. The fact that we have sexual selection in there is actually really important, because that always poses part of these answers for natural selection. So basically, the fittest, basically it's the idea of survival of the fittest. The fittest animal will survive. So if one has, if you've got one more successful squirrel that's better at getting nuts, via natural selection, it's going to survive and live for longer. Therefore, it's also probably more likely that it's going to reproduce more, and females are going to choose to mate with that male squirrel more because it has lived longer, it's fitter, it's healthier. It's that sort of, both of them come hand in hand. So, Charles Darwin, you have to know a little bit about his life. So we'll just have a little quick flick through this. So in 1825 to 1831, Charles Darwin went to university and he actually studied medicine at the University of Edinburgh. But he left his second year to study for the clergy, so to join the church at Christ's College in Cambridge. Whilst Darwin was there, he became fascinated by natural history and was a keen naturalist in his spare time. In 1831, so pretty much once he finished studying to be part of the clergy, 36, Darwin visited the Galapagos Islands during his time as official naturalist aboard the HMS Beagle. He made detailed notes about the animals he found and collected many animal skins, plants, insects, fossils, and shells that he brought back to the UK to study. In 46 to 54, this is when Darwin's early research really happened. So he went to the Galapagos Islands, made all this notes, he got all this evidence, and he basically brought it back with him, and then he sort of just sat and looked at it and decided, hang on, what does this mean? And he didn't just go to the Galapagos Islands, he actually travelled around lots of places, and Galapagos is one of the most famous places that he went. So after he returned home, Darwin began an 8-year-long study of barnacles. So barnacles are like little sea creatures that often will grow on the undersides of ships and on rocks. And he did this in order to establish his scientific expertise and to gather evidence before publishing his revolutionary ideas in evolution and natural selection. So if you think, he's gone off and done this, studying and travelling, but he actually studied at university to go and join the church. He wasn't a renowned scientist at this point, and he had these ideas and he needed stuff to prove and back him up, because otherwise he would just probably look like a bit of a crazy person. And he did to lots of people anyway. So in 1859 even to 1864, there's a lot of controversy. So in 1859, Darwin's book, The Origin of Species, went on sale. The book sold out and it was really, really well received in scientific circles. However, supporters of the church were outraged that Darwin's ideas contradicted what was written in the Bible. So then there was lots of controversy and lots of writing backwards and forwards and people trying to disprove Darwin and Darwin trying to prove himself. And in 1864 to 1882 was the life's work. So in Darwin's latest years, the controversy over his work decreased, and he was able to publish a book describing the evolution of humans to little outcry from the church. Today, Darwin's theory of evolution is considered a central concept in biology. So I'm going to look at natural selection. Again, nothing too new from GCSE, but we need to be really specific how we talk about it now. So Darwin's theory of natural selection is based on the fact that natural variation amongst organisms causes them to differ in their ability to survive and reproduce. And it's natural variation. This variation it's not directional, it's not been chosen, it's that random mutation. we have variation, all humans. We look around, everyone looks slightly different, and everyone's bodies work slightly differently. And in wild animals and wild plants, what we can see is those natural variations. Some of them are advantageous and some of them are disadvantageous. Those advantageous ones help them to survive and reproduce and will get passed on to future generations and can then direct evolution. Those more disadvantageous ones often don't get passed on because they don't help survival or reproduction. So competition. For natural selection to work, there needs to be an element of competition. Why? Well, basically natural selection helps an animal. Basically natural selection is this random trait that helps the animals survive and reproduce. If there is zero competition within the population, so every animal's got the same amount of food, ample amount of food, more food than it could possibly ever need, and there was no competition for mates, whether some rabbits, so from this example, we've got some rabbits having white fur and brown fur. If their white fur and brown fur made no difference to either their survival or their reproduction, you would just have continuously slightly fluctuating, but generally towards 50-50 amount of rabbits with white and brown fur. Obviously, depending on the dominant and recessive alleles, that might change, but you would have basically this constant flow of white and brown furred rabbits and it wouldn't change. For natural selection to occur, there needs to be competition. So animals have to compete for limited resources. These limited resources can be things such as food, shelter, mates, and they also have to avoid predators, which is a competition. The one who's better at avoiding a predator wins. So there needs to be, for natural selection to occur, there needs to be some element of competition. So adaptation. This is one of those random differences in their bodies that allows them to actually compete and be better. So let's look at those rabbits. We've got the brown-furred rabbits and the white-furred rabbits. If these rabbits were living in a snowy environment, rabbits with white fur will be better camouflaged than the brown rabbits, and therefore they are better adapted to their environment. Because they are better adapted to their environment, they are more likely to be camouflaged. Because they're more likely to be camouflaged, that means they are less likely to be seen by predators and caught by them. Therefore, more brown rabbits will get caught. So that's the adaptation in this situation. So for successful gene. So the white rabbits are more likely to survive than the brown rabbits. And therefore, because they're more likely to survive, they're more likely to reproduce. And this is where that natural selection and sexual selection comes in. If all the brown rabbits have been eaten, there's hardly going to be any around to reproduce, to pass on them genes, because they don't survive as well. So the successful gene, the white fur, will then be passed on to the next generation, but the brown fur genes won't. And that's literally because the white rabbit has survived and the brown rabbit hasn't. So what happens next? So as long as that environment stays exactly the same, the frequency of white rabbits in the population will gradually increase over time at the expense of the brown rabbits. What would happen if global warming caused the snow to melt? Well, we'd expect to see the exact opposite. Now, the successful gene would be the brown fur, and the white rabbits would be less successful, and the exact opposite would happen. So let's have a look at some natural selection in action. So the common tree snake is a native of Australia, and it feeds mainly on frogs and geckos. So over the past few decades, the snake has evolved a reduced head size. So the cane toad. In 1937, the cane toad was introduced to Australia, where it became a major pest. This actually happened quite a bit in countries like Australia. People would go and I can't think what the word is now. When you go somewhere and you sort of settle, the settlers would then take their own animals. So obviously the countries would have their own population of humans, they'd have their own population of natural wildlife and fauna, and people would go, oh, you know what would be really great? Let's take this random animal from a completely different country and bring it here. So it's like the grey squirrels here. Grey squirrels aren't native to the UK. Red squirrels are. We don't have many red squirrels now because the grey squirrels basically wiped them out. Grey squirrels came from America. So we get a few red squirrels now in Lake District in Scotland, but we don't get many. It's because the grey squirrels were better at surviving. So cane toad is one of those examples. It became a major pest in Australia. It became a bit of a pain. Cane toad is highly toxic, and its large size ensures that any predator eating the entire animal receives a huge dose of poison. So, selection pressure. Initially, the common tree snake fed on the cane toads. They hadn't encountered these toads before, so they had no resistance to its poison. So what happened was that most snakes that then fed on the cane toads quickly died. This exerted a really strong selection pressure. So again, this is another key part when we talk about natural selection, is a selection pressure on the snake population. The snake population was dying out because of these cane toads. Selection favoured any snakes that could avoid eating the cane toads. Now, snakes aren't going to go around with this lucid thought process going, oh, I'm going to eat a cane toad, I'm not going to eat a cane toad. It's used to eating frogs, sees frogs, going to eat it. Cane toads, the snakes that weren't going to eat the cane toads were going to be the snakes that didn't have big enough mouths to eat these massive cane toads. So you can probably see what's going to happen. Selective advantage. As snakes eat their food whole, a small head prevents them feeding on the large prey. Therefore, small head in this situation has a selective advantage. It allows the snakes to easily avoid cane toad poisoning. So basically what happened was all the snakes with really big heads died out because they ate these cane toads, and the ones with small heads that couldn't eat the cane toads survived. So therefore, you can imagine, because they're the ones that survived, they went on to reproduce, and their young also had quite small head size, that meant they couldn't eat a cane toad. Small head snakes had a greater chance of surviving to reproduce, so the average head size of the population shrank as more small-headed snakes survived to pass on their genes. So, conditions for evolution. There are 3 factors needed for a population of organisms to be able to, evolve. One is variation. There has to be differences between the individuals in the population. If they are all completely identical, whether it's behaviorally, morphologically, physiologically, if they're all identical, then evolution can't happen because we have to have that variation. We have to have that slight little bit of difference between them for something to be advantageous. We need heredity to be in there as well. So this is the differences between those individual organisms must be inheritable. So, for example, what would be an example? I don't know. Let's say a squirrel that had lost its tail actually found it was able to live for longer. That isn't going to help an organism to evolve because that has happened after it hasn't been born without that tail. It's lost its tail during its life, so it can't help. So if it does survive for longer, yes, it is going to reproduce, but it's not going to pass on that lack of tail because it wasn't born without a tail, it's lost it. So any variation in the individuals that have happened post-birth that aren't genetic-based, things that are heritable, aren't going to allow evolution to occur. And then finally, a means of selection. So there must be a mechanism or a pressure that selects some variables for the next generation at the expense of others. So if everything is equal, if everything is fine, so if there's no harsh conditions, if everything's got food, ready supply of mates, then there's again not going to be any evolution because there's no selection, there's no means of selection, there's no mechanism, there's no pressure. So it's why, for example, if we look at humans, humans now sort of don't really evolve because we don't have that mechanism or selection pressure the same as animals do, because we work and think in a different manner, and we look after people. So the idea is that obviously if we look after people, we can help people, so then therefore there's no selection pressures on those differences between us. What was that? So all species exhibit variation between individuals, but we can have discontinuous and continuous variation. So discontinuous variation is there's a limited number of traits and there's no intermediate form. So an example of that would be, I mean, you could even go, because variation can go down to sex, male or female. White fur, brown fur, with a tail, without a tail. Or we can have continuous, so range or gradation of characteristics. So it could be short tail, medium tail, long tail, height, weight, things like that. So discontinuous variation is controlled by a few genes, and it's usually with multiple alleles. Continuous variation is controlled by many genes, and we say it's polygenic. Discontinuous variation, the environment has very little effect on these characteristics. So for example, male or female. Continuous, the environment has a significant effect on these variations. So discontinuous, some other examples are things like human blood groups. They're controlled by a few genes. We have a limited number of these traits. The environment has very little effect on them. And again, antibiotic resistance. Opposite, for continuous, we would have stem height in plants. You know that the environment has a huge significant effect on that. The better conditions it is, so the more sort of sunlight, the optimum levels of sunlight, water, oxygen, carbon dioxide, nutrients in the soil, moisture levels, going to help the plant to grow. Same as sort of milk yield in cows. So genotype and environment can both determine the phenotype. So the origin of genetic variation. So genetic variation is heritable. It is the variation that natural selection acts on, and the causes of genetic variation are mutation or sexual recombination. So let's look at mutation first. So mutations, we've talked about mutations in the past when we were looking at some genetics. So mutations are when we're copying the genetic code, and we can have a deletion in there. So we might have one base unit deleted when we're going through I've lost the word for it. Protein synthesis, germ protein synthesis. We could have a deletion. We could have an addition. We could have a substitution. Any times that we're creating new cells will go through mitosis, and we're going, all the DNA is being replicated. Again, if there's any bases that are deleted, any bases that are added or substituted, we can cause a mutation. We can also have deletions or translocations of part of a chromosome. So when obviously our chromosomes line up down the middle of the cell during mitosis, and then the centromere is attached to them and they get pulled apart. Actually, some parts of them can get deleted, they can get broken off and basically but broken off and degraded, so they're deleted. Or translocation, we can actually get pulled apart. So when they're crossing over and linking, some parts can get stuck to the wrong part of the chromosome and they can get pulled apart then. So in mutation there, we can get aneuploidy, which is a loss or gain of a single chromosome, or polyploidy, which is the addition of whole chromosome sets. Or we can get genetic variation via sexual recombination. So remember, when meiosis happens, we have that independent assortment of chromosomes. So this is why brothers and sisters of parents who have the same parents don't look identical to each other, because actually those chromosomes that are there when they line up, you obviously get half from your mother, half from your father. They're not ever the same exact half, because they cross over. You get a little bit of this chromosome and a little bit of that one. So we have this independent assortment chromosomes in meiosis. We have crossing over during meiosis, and we have random fertilization, which creates that genetic variation as well. So the conditions for evolution. So a gene or a point mutation changes the base sequence of a gene. Mutations are caused by errors that occur during DNA replication. So we have to have this to occur. We have to have some sort of change in the genetic code. So mutations can be silent. They do not have an effect, and therefore they don't change the organism's survival chances. So you can have silent mutations. We can have harmful mutations, so these can reduce the survival chances of the individual, or we can have beneficial mutations, and these obviously improve the survival chances of the individual. Beneficial mutations are very rare, so that's really important to remember. This is why evolution is a long, slow process, because actually most mutations are either going to be silent, harmful, or in a really rare chance, beneficial. So mutations provide the genetic variation that is required for evolution. So we need these mutations to provide genetic variation. So the natural spontaneous mutation rate is typically around 1 or 2 mutations per 100,000 genes per generation. So the mutation rate can be increased by mutagenic agents. These include things such as UV radiation and harmful chemicals. These disrupt the structure of DNA or interfere with transcription. And again, we're using things like terms like harmful chemicals. These mutations that occur generally aren't going to be beneficial. So let's get the last bit of this. Selection pressures now. We're going to look at evolution and natural selection pressures. So types of variation and what selection pressures are. So selection causes some traits to survive and spread while others are lost. And what determines this is a selection pressure. So selection pressure decides what trait is going to be successful or not. And that's basically the basis of it. So we have 3 types of selection that you need to know about. So we have disruptive, stabilizing, and directional. And we'll look at them in a second. So selection, we can actually use graphs to represent this, and selection is able to show the distribution of individuals with a particular trait. So the number of individuals and the trait across the bottom, the starting population always has a normal distribution of traits. The mode, middle, and then either direction. So that's always our starting population. So selection can cause the mode from here and or our distribution to change. So this is a normal distribution here of variation of that specific trait. So we could have, as we said, disruptive selection. So disruptive selection creates this situation where we basically get 2 modes. We get 2 peaks, and it's where you've got 2 traits that are present, and we've got 2 dominant traits, and then everything else is moved away. So the selection pressure is actually going towards the extremes, and so we get these 2 modal values. So we've got as far away from each other as possible, and that's a disruptive pressure, and it creates these 2 extremes. So we have 2 modal values at the extreme, and basically that mode that would have been in the middle with that normal distribution is just gone completely. We can also have the stabilizing, so we get this. So the selection pressure toward the center actually increases the number of individuals at the modal value, so that area increases, and so actually some of them won't be exactly right on the mode where the trait is, that optimum, but actually we've got lots of them that are more towards that center, and we have a bigger drop off that's quicker either side. And then we can have directional. It doesn't have to be to the right, that directional could be to the left of the graph as well. And this is when selection pressure towards one extreme moves the mode in that direction. So we're going to have a look at some selection pressures now. So yeah, the 3 that you need to know are disruptive, and that's selection pressure towards the extremes, and we get 2 modal values. Stabilizing, selection pressure towards the center, increases the number of individuals at the modal value, and then directional, and the pressure goes towards one extreme. It can be either extreme. So how does selection affect a population? This is probably one that you've seen loads, and it is the moths. So when the mottled phenotype is the most successful, so I can't remember what these moths are called now, but you see them loads, and basically you get these moths, and they have basically 3 pigmentations. And you could have white, you could have black, and then you could have these mottled ones that are in the middle. And the whole sort of story behind them is that there was sort of there was a higher distribution of these white moths. The Industrial Revolution came along, and basically then you'd got the, let me go back. So you had a high amount directional, so it was towards the white moths. Then selection pressure came along, Industrial Revolution, and basically these moths lived on like silver birch trees, so they blended in with the colour of the bark. Industrial Revolution came along, so basically went on to everything went really dark. The black moths survived a lot better because obviously they camouflaged in a lot better with the trees. So then it was directional pressure again to the opposite end. And then we actually can get this mottled one to look at in the middle. So we'll look at this in a bit more detail now. So when the mottled phenotype is the most successful, the number of individuals with this phenotype increases. Is this going to play? Yes. So when this happens, this is a stabilizing selection. So this causes a distribution, I'll play that again. This causes a distribution of the phenotypes to become more heavily clustered around the center. So that would have been, then we've got directional. When the pale phenotype becomes the most successful, so let's say you've got loads of silver birch trees, the number of individuals with this phenotype increases and it goes directional to the left. This causes our modal value to move towards the pale phenotype, and the distribution of phenotypes become clustered around the new mode. This is directional selection. This could also happen in the opposite direction, like I was saying, during the Industrial Revolution, and that would have actually happened and gone the opposite way towards dark moths. And then obviously we've got disruptive selection. When the pale and dark phenotypes become the most successful, the number of individuals with both of these phenotypes increase. This creates our two modal values, and the distribution of phenotypes becomes clustered around the two new values, and this is our disruptive selection. So humanity as a selection pressure. So human activity provides some of the strongest selection pressures in the world today. So we're going to have a little look at antibiotics. This is a major evolutionary selection pressure sort of creation, if that makes sense. So the widespread use of antibiotics has exerted a very, very strong selection pressure on bacteria. So any bacteria that can survive exposure to antibiotics can rapidly divide and reproduce a resistant population. So basically how this works is, bacteria reproduces using binary fission. So they basically split down the middle and they duplicate very, very fast. If they're the only ones, so bacteria reproduces quickly. If you've got some resistant individuals in your population to the antibiotics that are being used, they can rapidly divide and produce a resistant population very, very quickly. That is issues with bacteria. They divide and produce populations quickly. So any that resistant really produce quickly. So MRSA is an antibiotic-resistant bacterium. So what kind of selection is this? It's going to be disruptive. Okay, it's going to go towards one direction. It's going to go towards the antibiotic-resistant bacteria. What are the implications of the evolution of antibiotic resistance for humans? It's not very good, to put it bluntly, because bacteria can reproduce it quickly. Any that are resistant are going to become the stronger, more dominant ones. So then it's why we have to try and look for so many more antibiotics. It's why we're trying to not use antibiotics too much anymore, because actually the less we use antibiotics and the more we let our body try and fight off bacteria ourselves, the less likely we are for these few resistant ones that you might have in your body to actually have that chance to create this this resistant population in your body. So if you think, for example, you've got bacterial infection, bacterial tonsillitis, if you're healthy enough and your immune system's good enough in your body and you're not too poorly, your body can fight it off itself. Your body's just going to kill off all that bacteria. It's going to kill it off all the same. It's not going to kill off the non-resistant stuff first or resistant stuff first. It's going to kill them all off. You might be a bit more ill for a few more days, but that's sort of generally how it works. If you took some antibiotics, but you had some resistant individuals in bacteria in your tonsils, for example, all those non-resistant ones will get killed off. then you've got all those resistant ones left, and they multiply and spread really, really quickly. So then you've got this resistant population. And obviously then if you're coughing and stuff around other people, you might feel better because most of those bacteria have been killed off in your body. So you're like, oh, I feel better, my antibiotics worked, you go off, carry on living normally, and it gets spread, and then we have a spread of this resistance. So it's sort of the idea that we're trying to rely a lot less on antibiotics now. It's also how we can get resistant strains occurring if people don't take the whole course of antibiotics, and a lot in there as well. Human birth weight, this is quite an interesting one to look at for selection pressures as well. So human birth weight falls within a narrow range of values, and that's between 2,500 and 4,000 grams. The mortality rate below and above these values are very, very high, and so it favours a narrow range of birth weights. What might cause the higher mortality rate in underweight and overweight babies? Or if you look at it, if you think about the underweight babies, that high mortality rate is going to be possibly because they're not going to be fully developed, they're not going to have, they might be premature, they might not have fully developed organs, so they're not going to have as good an immune system, the body's just not going to be working as well. Babies that are larger, the high mortality rate, that's actually going to be coming in to probably more about in labour and during birth and how the danger's there. So how might a caesarean section and improved care for premature lighter babies change this distribution? Well, the idea is that caesarean section, a natural birth can be quite dangerous for a very, very small baby. It's less likely to survive. And if we can improve care for these premature lighter babies, so in things such as neonatal intensive care units, they can survive for longer, then actually that graph distribution is going to change, because actually that low mortality rate is going to decrease, its range is going to increase and go into that low birth weight zone. So you're actually going to then potentially have more babies being born that are of lower birth weight, because some people just naturally give birth to babies that have a lower birth weight. And if they survive, then they themselves, when they go on to have a baby, are getting more likely to have a baby that is of a lower birth weight. Okay, so let me pause the recording, or let me stop recording actually. Right, stop share, that'll be better.